So hello everyone. My name is Dionne, and today I'll be talking a little bit about my research. So an investigation of K2 Mycobacteriophage Lysin A proteins. So just to give a little bit of background, so mycobacteriophages are viruses that are specific to mycobacteria. So things such as Mycobacterium tuberculosis, um, are particularly relevant as they're a challenge in the world of infectious disease, where they're very pathogenic and becoming antibiotic resistant. So one way that we can use phages as an alternative to antibiotics. So if we can use these viruses that are specific to these infectious pathogens and kill the bacteria that way, we might be able to use these therapeutically. So K2 mycobacteriophages are simply a subcluster of genetically similar mycobacteriophages that have a broad host range, and they make them particularly attractive to be used for phage therapy, is what we call it, is the therapeutic application. So with my research, if you'll go to the first figure on the bottom left, we have something called an immunity assay, which involves creating bacterial lysogen. So first off, the strain of bacteria that we use in the lab is not Mycobacterium tuberculosis. That would be kind of dangerous. We use a non-pathogenic strain of bacteria called Mycobacterium smegmatis. So that's the bacteria on those plates there. And with an immunity assay, what we do is we've taken these few K2 mycobacteriophages, so Millie, Mufasa, TM4, and Zoe J. And we plate them against a lawn of Mycobacterium smegmatis, and we observe their infectivity. And you can see that all of these phages infect the bacterial lawn very strongly with those dark circles there. But then what we can do is we can isolate a lysogenic strain, which has incorporated a small portion of the viral genome and is therefore resistant to infection by the same phage. Um, so you can see here in the bottom plate, we've made a milli lysogen, so it's incorporated part of the milli viral genome. And we can see that milli has no infection of that bacterial lawn, whereas some of the other phages do kind of infect. With Zoe J at the bottom, we see kind of a partial infection of the bacterial lawn, which we would normally expect with genetically similar phages to see some reduced infectivity, whereas TM4, which is actually a lytic-only phage, meaning that it's not affected by lysogens, we see full infection comparable to our control here with just normal smegmatis. However, with Mufasa, we noticed something a little weird, where we saw no decreased infectivity despite this phage having the same lifestyle as Zoe J, where we would normally expect some kind of decreased infectivity here. So this was just one of the experiments that we did and reproduced in our lab that kind of suggested Mufasa was a little kind of anomalous in some of the phenotypes we were seeing. Another experiment that I did with Mufasa is, I kind of alluded to this a little bit with the immunity assay, but there are two different phage lifestyles. So there's a lytic only, which is what we see in TM4, where even in the presence of this lysogen, we don't see any decreased infectivity, which, if you can imagine, is what we want when we're talking about a therapeutic application. We want these phages to be killing all of the bacteria for treatments. But what we can do to make some of these phages exclusively lytic only, so that they kill all of the bacteria, is delete a particular gene in the genome called the immunity repressor. So that's what we're doing currently, and what I've tried to do with Mufasa is to delete that gene. And one way we can do this is using a technique called bacteriophage recombination with electroporated DNA, or BREAD for short. So what we do here is we create a substrate of DNA that contains sequence homology upstream and downstream of the gene we want to delete. And then we can shock the cells using um, uh, with a recombination plasmid that has all of the cellular mechanisms needed for this recombination event. And we end up with a deletion kind of a reorganizing of the DNA to get this deletion of the gene that we want between our substrate. And so we can run this on a gel to confirm whether or not the deletion is there, and the presence of a band on the gel suggests that our deletion was successful. So that's what we see on this gel here with this 223 base pair amplicon in figure 2. Um, a purify mutant will do multiple stages of screening. This comes from a primary screen. So we were able to detect the mutant with that missing immunity repressor in the primary screen, but subsequent screens, we weren't actually able to purify this particular virus to any more stages. So all of our screening was negative to the second and third stage. And to troubleshoot this, we tried a number of different things, including different primers, like starting the process all over again with another electroporation, multiple, just going back and doing PCR again. And we couldn't actually get it to a second or third stage of purification. So this result led us to believe that this particular mutation in Mufasa was not viable, which was strange when we consider that doing it with other K2 phage that we have in our lab, we were successful with this same kind of deletion. So this is yet another reason why we think Mufasa, something's going on that's different here. One thing we wanted to kind of confirm to rule out anything that's going on with this immunity uh, assay here is we wanted to make sure that our Mufasa sample wasn't contaminated with a different kind of phage. That would mean that it would still show this infectivity even on this millilysogen. So what we did here is we kind of tried to analyze its DNA of the sample. So what we can do is we can expose the phage DNA to a nuclease enzyme, which cuts up the DNA at a specific site. And we compare the DNA pattern that it makes in the gel. So you can see here, Mufasa and Zoe J are both very genetically similar. So we expect a very similar band pattern in the gel. And that's what we do see here with these two. And we just have some different controls. This is an E cluster, which is very genetically different, and then a B1. And you can see how those band patterns are different. So what we kind of proved with this, or confirmed, is that our Mufasa sample is at least a K2 subcluster phage. So there's no kind of contamination resulting in this still very high infectivity of the lysogen. So kind of moving on from these kind of anomalous results with Mufasa, we wanted to see and think about what else could be involved in this infectivity that we were seeing. And an analysis of the genome, which you can see up here in figure 4, this is kind of a genome schematic map where each of these different rectangles represents a different open reading frame, which symbolizes a different enzyme or protein that's being expressed. So a genomic kind of analysis bioinformatically led us to believe that maybe the lysin A was involved. So a lysin A is a type of endolysin, which is responsible for breaking down the peptidoglycan, which is a layer of the cell wall that's necessary for the cell to remain, to have structural integrity. So we can see here that the boxes between Millie and Zoe J, this magenta box, this represents a particular lysin A. And then with Mufasa and TM4, this lysin A here contains some different domains and belongs to a different kind of family here. So something that I wanted to investigate is maybe this different domain that Mufasa and TM4 share that other K2 phages do not has something to do with this infectivity phenotype that we were seeing. So what I kind of sought out to do is to isolate the lysin A protein and maybe get like a structural analysis, get a structure of it that I could kind of biochemically characterize to see how it might be different structurally and what kind of impact that might have on the function of cell lysis and the cell death that we're seeing with the clearing of the bacterial lawn. So to do this, this kind of cloning of the Lysin A to isolate it and purify it, I needed to make primers that contain regions of the Lysin A that overlap with an expression vector plasmid. So in this case, I'm going to use the PET28A expression vector. And from there, I'm going to use these primers to create and amplify an insert. So an insert that has overlap with the expression vector plasmid and the full gene that encodes the Lysin A. And then what I'll do is I'll transfer this and use that overlap with the expression vector to put it on the full plasmid, which I'll then transform. Um, sorry, which which I'll then synthesize. So I'll integrate the insert into the expression vector and then transform this into cells that can copy the plasmid for me. So now I have a bunch of copies of this expression vector plasmid with the gene that I want. And then I can then transform that again into cells that will synthesize the Lysin A protein for me, and then isolate that using different protein purification methods such as affinity chromatography. So where I'm kind of at in this process, I'm not really done yet, so I haven't gotten the structure or protein. But what I have here is I have a linearized empty expression vector plasmid, and I have my amplified insert with the sequence homology ready to be inserted into this plasmid. And from here, I'll do a process that's called Gibson assembly, which is where you actually integrate the gene into the expression vector. And then from there, I can continue with the protein purification. So yes, that's kind of where I'm at right now. Uh, I just kind of received funding very close to the end of the semester, so I haven't really had a chance to move forward too much with this. But thank you thank you very much for listening to my talk, and I'll take any questions you might have.
Yeah, thanks so much. I really appreciate it. So we are a small group, so feel free if anybody has any questions or comments to ask them in whatever format you feel most comfortable, chat or just shouting out.
Ethan, I'm just going to shout out here. Is that okay?
Yeah, that's fine.
Great talk. A couple of questions. So have people isolated these lysins before and characterized their antibiotic activity?
Yes. So that's something else that we wanted to check on too, getting a Lysin A structure and a protein sample. Is that these lysins have actually been shown to have some kind of antibiotic activity. Um, while it's not always a strong kind of antibiotic quality, they have been shown to inhibit the active growth of bacteria. So there is some chance that these lysin proteins can be used themselves as a therapy.
But I guess your research is showing that these Mufasa and Team 4 have hyperactivity, and so you're hoping that maybe there's something about this lysin protein that's better. Is that true? Is
Yeah,
that accurate?
yeah, and it's also about understanding this particular domain that's different between the different phages here.
Another question I had was if you look at figure 1, so if you notice like in Millie and in TM4, like the third column over, it has this white spot inside, and same thing happens for Zoe J on the bottom row. What is that? Some impurity or
Yeah, I
infection or
believe that's a little bit of contamination on the plate, so that's not actually significant.
Okay.
Yeah. These were fairly old plates that I photographed. Or I didn't actually photograph those. They were kind of,
And this is a dilution series, is that correct?
yes, this is a dilution series.
Okay.
So the most concentrated is on the left and the most dilute is on the right.
Thanks.
Yeah, thank you. For a non-biochemist like me, can you say a little bit more about what you hope to learn from the structure of this lysin protein? So what will knowing the structure allow you to do or allow you to know that you don't already know?
Yeah, so knowing the structure of a protein can tell you a lot about its specificity, and the kind of reactions or different things that it can interact with. Um, so kind of knowing the structure between uh, the two lysins, between something like Zoe J and then Mufasa, this like comparative structural study can tell us maybe more about the kind of reaction it does, how efficient it is at breaking down peptidoglycan to lyse the cell. So that's kind of what I hope for. There are a lot of challenges to getting a structure of a protein, depending on, what the protein does and where it's found.
I can only imagine.
Is this a membrane protein or is this a freestanding like enzyme?
I believe it's a freestanding enzyme. It's
Cytosolic, some sort of
not a membrane.
cytosolic protein.
Yeah, so endolysin means that it functions from the interior of the cell.